Hello everyone! Today we want to talk about biochemical signaling. So here's my outline for today. We want to talk about an introduction to cell signaling using gases and hydrophobic signaling molecules as examples. And later on we'll talk about receptor tyrosine kinases, the heterotrimeric G proteins, and then the phosphoinositide pathway. But today is mainly an introduction. So first, what are some of the principles of cell communication? Well, we are multicellular organisms, so our cells need to communicate with each other. So we have signaling molecules. We can call them ligands or agonists, and they bind to receptors. The receptors can be outside the cell, or they can be inside. And when the ligand binds to its receptor, it generates a cascade of reactions that alters the cellular behavior. So this just shows very sort of generically. So here are my signaling molecules right here shown in purple. They diffuse out of the cell, bind maybe to a cell surface receptor, and then the cell surface receptor puts into motion a cascade of reactions that causes some kind of effector protein to be activated, which could lead to modifications of cellular metabolism, function, movement, gene expression, development, lots of different things. So I first wanted to just go through a couple of examples of different types of signaling. So this is called contact-dependent signaling. So what's happening here is that the signaling molecules remain bound to the surface of the cell. So that means the only cells they can influence are the cells that contact it. So if this is the signaling cell over here on the left, and these little purple things right here are the signaling molecules. When they bind to the receptors, they can only do so if the target cell is right next to it. So contact-dependent signaling is important in the immune system and during development. And of course, we can take whole courses just in those fields. I know some of you are interested in neuroscience, so this would involve synaptic signaling. So these molecules are secreted and may be carried far afield to act on distant targets. So if we have our neuron right here and a very long axon, then the signaling molecules right here are the neurotransmitters that are shown in red. And then they leave the presynaptic cell and then bind to receptors on the target cell. And again, when they bind, they cause certain effects on the target cell. So for the synaptic signaling, these axons can be very, very long. So neurons send action potentials along the axon, and this causes the nerve terminals to secrete a chemical signal, the neurotransmitter. And again, the neurotransmitter is shown as this red circle right here, and that's delivered to the postsynaptic cell in the form of the neurotransmitter binding to the receptor and then causing a change in the target cell. So here this shows a cartoon of the different neurons being able to influence various target cells. So endocrine signaling, so here these signaling molecules are secreted and then they're carried far afield, and here they're carried by the bloodstream. So we have an endocrine cell that secretes a hormone, and then that enters the bloodstream, and then the bloodstream carries the hormone to bind to its target cell. So again, there will be certain cells that will have receptors that can bind to that ligand, and other cells won't respond to it if they don't have that receptor. So this shows here various endocrine cells that are secreting red circles or blue squares or green triangles, and the target cells will only respond to the signaling ligands, the hormones secreted by the endocrine cells, as long as they have the receptors in order to be able to bind to those signaling molecules. So this is just sort of a generic explanation of what happens. So if we have an extracellular signaling molecule and it binds on a cell surface receptor, then that activates some kind of intracellular signaling pathway that can alter protein function. Maybe it alters cytoplasmic machinery, which causes a change in cell behavior. Or maybe it's able to influence what goes on in the nucleus in terms of binding to DNA and ensuring that messenger RNA and protein is made. So we have altered protein synthesis, which also can alter the cytoplasmic machinery and alter cell behavior. But the time scale is different here. So there are some very fast pathways that act within seconds or minutes. But if we're influence the synthesis of DNA to RNA to protein, that's going to take much longer. So in the synaptic versus the endocrine signal, they both they're involving targets that are far away. But since endocrine signaling relies on diffusion and blood flow, it's going to be a whole lot slower than synaptic signaling. So of course, for the nervous system, we want our target cells to be able to react very rapidly to the binding of the neurotransmitter to the receptors on the target cells. And the reason they can do that is because this synapse, this space between the neuron and the target cell is very, very small. So the signaling molecules become very, very concentrated. Whereas for endocrine signaling, the signaling molecules become very dilute in the blood. So it will be a lot slower for these signaling molecules to find their targets. Here's another kind of signaling called paracrine signaling. So here, these signals they can act over short or long distances. That here, it is showing a short distance. So they might act as local mediators. So we have a secretory cell secreting some kind of target that is acting on a cell that's nearby. We can have also autocrine signaling, which means that the target site is the same cell. So the cell that's secreting the ligand is also it involves binding to the receptor. This is used by tumor cells. So I think I showed you this slide when we were talking about apoptosis. So there are certain signals that cells need to interpret. There could be survival signals, growth and division signals, differentiation signals, and if there are no signals, that's when the cell undergoes apoptosis. So if you think of surviving, growing, dividing, differentiating, you can see why people are interested in this, because cells that survive longer than they're supposed to, or grow and divide more than they're supposed to, or differentiate less than they're supposed to, those are all the hallmarks of tumor cells. So you can see this is why people are interested in this field. So it's interesting that different cells respond differently to the same signal. So how a cell responds to a signal depends on what receptor protein the cell has, and it depends on what the intercellular machinery is, because that's when it's going to integrate and interpret that signal. So here are a couple of examples. So acetylcholine, for example. So here's the structure of acetylcholine. And again, it's shown as this red circle. So if this is a heart muscle cell, then the acetylcholine binds to its receptor. And the response to the target would be decreased rate and force of contraction in the heart muscle cell. But that same ligand, that same agonist, acetylcholine, when it binds to a skeletal muscle cell, will open up a gate and cause the muscle to contract. Or for a salivary gland, then that same acetylcholine will bind to its receptor and cause the secretion of salivary enzymes that will help digest our food. So same ligand, but different cells, different receptors, and different ways of the cellular machinery integrating and interpreting the signal that it's receiving. So the response will fade when the signal ceases. We don't want these signals to be on all the time. So the turnover rate determines the promptness of the response when the signal is turned on. So what we want is we want these molecules to have really short half-lives. So if you have a molecule with a short half-life, that's going to have not just a more rapid boost in concentration when the signal is turned on, but it's going to be a more rapid decline when the signal is turned off. So it's going to be a very, very sharp on-off switch. So most signaling molecules have short half-lives, and that's demonstrated on this next slide here, where you can see if you have a molecule with a half-life of a minute. If you look over here for part B, so this is on the x-axis, this is the minutes after the synthesis rate has been increased by a factor of 10. You can see we can really ramp up the synthesis of this molecule, and we can also decrease its amount when the signal has passed. Whereas if we have these molecules that are shown in green, with much longer half-lives, then it takes a lot longer to get your signal and it takes a lot longer to turn it off. Okay, so let's go back to our outline, and we want to talk about introduction to cell signaling. So today we want to talk about gases and hydrophobic signaling molecules like steroids. So let's talk about gases first. So gases as signaling molecules, this is kind of a recent innovation because you can appreciate that this would be very hard to detect. So, NO is actually a signaling molecule. So we have an activated nerve terminal here, which secretes acetylcholine. And then the acetylcholine binds to its receptor and activates NO synthase or NOS. So that enzyme then acts upon arginine as a substrate and produces a gas, NO. And because NO is nonpolar, it diffuses across the membrane and then it can diffuse into a neighboring cell, and then it binds to a soluble form of guanylyl cyclase, and that converts GTP to cyclic GMP, and then that acts on its targets to relax the smooth muscle. And we can terminate the signal if we have a cyclic GMP phosphodiesterase that will hydrolyze the GMP, the cyclic GMP to just regular GMP, which is not a second messenger. So you might be interested to know that Viagra is a cyclic GMP phosphodiesterase inhibitor. And in fact, a lot of drugs are inhibitors of one kind or another for some of these signaling pathways. So again, you can see why people are interested in these pathways. So more about NO. I believe that this was used as a treatment before people knew how it worked. So this is nitroglycerin, and it's used to treat angina or chest pain that occurs when there's insufficient blood flow to the heart. So you might have, I don't know if you have experience or know about this, about of taking nitroglycerin tablets and put it below the tongue, because that can speeds the absorption of this compound, and the nitroglycerin is then converted to NO, and then that relaxes blood vessels and that decreases the workload on the heart muscle. So I should have looked this up before, but I believe what is that people use this before they actually knew the mechanism of the NO signaling mechanism. So here's the chemistry of how this works. So this is in our book. So here is arginine, and we need oxygen and NADPH also as substrates to convert the arginine to L-hydroxyarginine. And then our compound, our products right here are citrulline, and here's our NO. So it's actually a free radical. Again, it is a gas, it's very small, it's nonpolar, and it diffuses across the cell membranes. Its half-life is about 5 seconds, so that was why it was hard to discover these pathways. So it doesn't go much farther than a millimeter away from its site of synthesis because it's just not around for very long. But this explains how it could work. So acetylcholine could bind to its receptor. Say it was a G protein coupled receptor, and we'll talk about those later. And it could activate an enzyme called phospholipase C, which produces IP3, and we'll talk about this later too, but that binds to gated channels on the ER and causes calcium to flood into the cytosol. And then calcium calmodulin then will activate the NO synthase. And then again, as I showed you before, arginine and oxygen then are the substrates to form a citrulline and NO. And then here, this shows the NO diffusing into neighboring cells. So if it diffuses into these muscle cells that underlie the endothelial cells in the blood vessel, then the NO can bind to its cytosolic NO receptor. Again, the NO just goes across this membrane. It's nonpolar, so it can easily get across. Binds to this NO receptor in the cytosol, and that activates the enzyme that converts GTP to cyclic GMP, and that activates protein kinase G, and that helps relax the muscle cell. Okay, what about other hydrophobic signaling molecules like steroids? So let's talk about those next. So hydrophobic signals are like NO, they're hydrophobic and they can diffuse across the plasma membrane, and they don't need a cell surface receptor, so their receptors will be inside. They can be in the cytosol, or sometimes they're actually in the nucleus. So steroid hormones, thyroid hormones, retinoids, vitamin D, all of these hormones act in this way. And the way that they work is they bind to DNA and regulate the transcription of specific genes. So this is just sort of a list to convince you that these lipid-soluble hormones are very physiologically important. So the adrenal cortex produces at least 50 different adrenal corticosteroids, and then and they can be classified in this way. So glucocorticoids affect carbohydrate, protein, lipid metabolism. So mostly they are the stress hormones, so they influence our ability, our capacity to cope with stress. Mineralocorticoids function to regulate the excretion of salt and water by the kidneys. And androgens and estrogens affect sexual development and function, and they're made in larger quantities by the gonads. So again, you can take a whole course just in studying these molecules. So these lipid-soluble hormones diffuse through the plasma membrane, and again, sometimes there is a cytosolic receptor, and sometimes they go all the way to the nucleus before they encounter a receptor there, and then they interact with transcription factors that they can control the expression of specific genes. So, so these transcription factors that act this way constitute the nuclear receptor superfamily. So hopefully some of us will look familiar because we, earlier in the course, talked about proteins that are able to bind to the DNA to influence transcription. So here are some structures to show you. So here's cortisol is a glucocorticoid, again involved in the stress response. Testosterone is an androgen, aldosterone is a mineralocorticoid, and beta estradiol is an estrogen. So they all have different ways of binding to the receptors and influencing the transcription of specific genes. Vitamin D. So you maybe don't often hear of vitamin B being a steroid, but it is. There is a precursor that's synthesized in our skin, and when our skin encounters ultraviolet light, is able to carry out this reaction to synthesize vitamin D. And vitamin D is important in promoting the uptake of calcium and reduces the excretion of calcium in the kidneys. So vitamin D and calcium metabolism go together. Okay, so if you are a hydrophobic signaling molecule, you're not going to be very soluble in an aqueous medium like the blood. So hydrophobic signaling molecules are bound to carrier proteins like albumin. So we've seen this before in nutrition where albumin will bind fatty acids, for example, because fatty acids are detergents, and having them be in the blood would be very bad for cellular membrane. So albumin really is a really important protein. So it not only carries fatty acids, but lots of these different kinds of hydrophobic signaling molecules as well. So what happens is that the ligand then needs to dissociate from the carrier protein, and then it can, again, go right across the membrane because it's nonpolar. It doesn't need any carrier to do that. And it may even diffuse across the nuclear membrane or go through those gates to encounter its intracellular receptor protein in the target cell. And then that will be a transcription factor. So a lot of these proteins have been studied. So, so these proteins right here are just lined up to show the different domains of the protein that have similar characteristics. So in purple, for example, that's the ligand binding domain. And these are for things like the estrogen receptor, progesterone, glucocorticoid, thyroxine, that's thyroid hormone, retinoic acid, that's vitamin A. And then this is just sort of the general primary structure that we're using to refer to all of these. So in blue, we have a DNA binding domain, and then there's a variable region depending on the type of ligand. So here's a picture right here. So the DNA backbone is shown in blue, and these alpha helices are shown in purple, and beta sheets right here in light blue. So so this is a C4 zinc finger. So the gray little balls right here are the zinc. So this is the glucocorticoid receptor protein. So it doesn't show this here, but the ligand will bind to the protein, and then that allows the protein to bind to the DNA in the major groove. And you can see that if you, I don't know if you remember that the zinc figure was one of those structural motifs that we find in proteins that have the ability to bind to DNA. So this might be a little bit more detailed than you're interested in, but remember, if and I have another slide to remind you that these proteins can bind as either homodimers or heterodimers. When we use the term response element, that's the sequence on the DNA, and that's where the receptor binds. So this would be the glucocorticoid receptor element. This is the estrogen receptor element. So again, if these are homodimers, it makes sense that if you look at the DNA sequence really carefully, that you'll see that they're inverted repeats, because if you have these homodimers, you'll have one dimer binding to one section of the DNA and one binding elsewhere. So we also have receptor elements that bind heterodimers. So these are direct repeats. And these are examples of the receptor elements that are able to bind as heterodimers. And here we have the direct repeats. And here we can show the arrows going in this direction that show the sequence that these proteins are able to bind to. So if that had a lot of detail, this is a little bit less detailed. So these are homodimers. So again, the homodimers would be identical dimers, and that means that the part of the domain of the protein that contacts the DNA, that the DNA sequence has to be similar. So that's what we saw in the previous slide, that we saw those inverted repeats. The heterodimer means we have two different proteins, and that means that they're going to be recognizing different DNA sequences. So the example that's shown in our book here is this is the glucocorticoid receptor binding domain. So you can see that here's one monomer right here shown mostly in pink, and then another one here shown kind of in a rainbow color right here. These are zinc fingers. So you can see that that cyan is the zinc, and you can see that these are the sulfur chains. I don't know if you remember that from a long time ago, that the sulfur chains form these coordinate covalent bonds with the zinc, the artist shows here the proteins binding in the major groove of the DNA. So here are the different kinds of receptor proteins, and they, of course, will all have a DNA binding domain, irrespective of the differences among these receptors. This looks sort of like a cartoony kind of thing right here. There's the ligand binding domain right here. This is the DNA binding domain shown in green, and that's the transcription activating domain that's going to interact with the RNA polymerase. So that's the inactive receptor. The active receptor, you can see there's quite a huge conformational change that occurs. So here's the ligand binding domain. So there's this conformational change that occurs, and then you can jettison the inhibitory protein, and then that allows the DNA binding domain on the protein to bind to the receptor binding element on the DNA. And often these proteins will attract other coactivator proteins too, and then as a complex they can interact with RNA polymerase and other proteins involved in transcription to activate transcription. So here is another example of the dramatic conformational change that can occur. So the ligand is shown here in red, so that's meant to show the steroid molecule. And what happens is the alpha helix that's shown here, it's just shown as a cylinder, but that's an alpha helix, and it snaps shut when the ligand binds, and it traps the ligand in place. But you can see that there's this huge conformational change that occurs. So prior to the ligand binding, this can't bind to the domain, but afterwards, the conformation is such that it will be able to do that. So this nuclear receptor superfamily, so again, some of them are homodimers, some of them are heterodimers. So the heterodimers, so in the absence of the hormone or the signaling ligand, the heterodimers bind to the DNA and they repress transcription by deacylating histone proteins. So you remember that when we acylate lysines, we got rid of the positive charge. So if you deacetylate the histones, if you deacetylate the lysine residues on the histones, you bring back the positive charge. So that means that these proteins are able to bind more tightly to the DNA. In the presence of the hormone, the ligand diffuses through the plasma membrane and the nuclear receptor and binds to the receptor, and then that leads to conformational change that results in hyperacetylation. So remember we talked about hyperacetylation, that removes the positive charge. So now the DNA becomes more available to the transcription apparatus, so the genes are transcribed. So this kind of signaling pathway takes a long time, and there's actually a primary and a secondary response. So the steroid hormone, when it binds to its receptor, will activate what's called the primary response gene. So will induce the synthesis of a certain number of genes, but then these will, if I look at the next slide, will act as transcription factors themselves and then turn on the synthesis of a secondary response. So you've got a primary response and then a secondary or delayed response to the steroid hormones. Okay. How about signaling molecules that bind to cell surface receptors? So we want to spend a lot more time talking about that. So we'll just introduce it now. So there are three different kinds: ion channel linked, G protein linked, and enzyme linked. So those are the three big categories of signaling molecules that bind to cell surface receptors. And these have short half-lives, unlike the steroids that have the long half-lives. So here, this is maybe we're more familiar with this because we talked about examples of this in Biochem 1. Our hydrophilic signaling molecule has no trouble being carried through the bloodstream, but it can't go through the membrane, so it needs a cell surface receptor. And then that receptor, when the ligand binds, has to communicate to the inside of the cell the fact that this hydrophilic signaling molecule has bound. So just to give you examples of these three categories. The ion channel coupled receptors, what they do is when they bind, they open up ion channels. And then so ions are able to flow through a cell membrane. So this changes ion permeability and then the excitability of the postsynaptic cell. So this is involved in nerve transmission. So I'm sorry this is not covered in this course because I know a lot of you are interested in neurotransmission, but that's the kind of receptors that they activate. The G protein coupled receptors you're more familiar with because this is our glucagon example. So we have this is a trimeric G protein coupled receptor. So here's our receptor right here. So that's the receptor that will bind the signaling molecule. And this is a trimeric G protein because there are 3 subunits. So the signaling molecule binds to its receptor, and then what happens is that receptor is associated with the G protein, and what happens is we have 3 subunits, alpha, beta, and gamma, and usually what happens is that gamma and beta, the beta and gamma subunits dissociate, and then the alpha subunits then activate an enzyme. So the example we're familiar with is adenylate cyclase, and that will catalyze the conversion of ATP to cyclic AMP. But there are other activated enzymes that are associated with G-coupled protein receptors. Adenylate cyclase isn't the only one. This field won the Nobel Prize in chemistry in 2012. A professor and one of his graduate students who also became a professor, and they together won the Nobel Prize for their work in this field. And I got to hear both of them speak at a meeting not too long ago. So the third category are these enzyme-linked receptors, and the way they work is they are single-pass transmembrane proteins, and they function as enzymes when they are activated themselves, or they might be associated with an enzyme. And it's usually a tyrosine kinase. So the way that this works is that these domains are inactive, but when the signaling molecule binds, the signaling molecule is in the form of a dimer, and when it binds, it dimerizes the receptors. And again, either it's an enzyme itself or it's associated with an enzyme. So here, this shows an enzyme itself, and this shows this protein, when it dimerizes, is associated with an enzyme, and then that enzyme can phosphorylate different proteins in the cell, which lead to downstream effects. So in general, the way these things work is they work by cascades. So you have a signal at the cell surface, and then there are a large number of proteins that are produced in sort of a cascade of reactions. And they can have all of these roles. So they can be relay, messenger, adapter, amplifier, transducer, bifurcation, integrator proteins, or latent gene regulatory proteins. So there are all kinds of different possibilities. So, so this shows what could happen. This again is just sort of a generic way by how these pathways work. Is we have the extracellular signaling molecule binds to its receptor. There could be scaffolding proteins that bring proteins together, so that sort of relays the message. And then we could have an enzyme that produces some kind of small molecule, and you could have one enzyme synthesizing lots of them, so that would both transduce and amplify the signal. And they bind to their own set of proteins. Maybe there are other signaling events in the cell that come together to integrate this signal, or maybe you can spread to other parts of the cell and spread the signal that way. And it is, again, possible that some of these might be involved in anchoring or modulation of activity, or others that can go into the cell, and like the steroid hormones, these proteins can go inside the cell and bind to some kind of response element or sequence on the DNA, and then have some effect on gene transcription. So the cascade part of this is really important, and that's what's being shown right here. So if we have, say, epinephrine, its concentration is 10 to the minus 10th molar, then one epinephrine can activate many adenylocyclase molecules. And then the adenylocyclase can synthesize many more cyclic AMP. So now we've amplified this by 4 orders of magnitude here to activate protein kinase A, and then protein kinase A can phosphorylate lots and lots of targets. So it all starts really with one signaling molecule. So you get one signaling molecule that amplifies the signal. So you have a small signal, but you get a very large response. So that is our whirlwind tour through cell signaling. I hope you enjoyed it, and I will see you next time.